Good morning, Rose.
Good morning, sir.
You know, again, one-on-one tuition here.
So it seems.
Okay, let's get busy. All right, let's get the chat up just in case anyone else pops in. Move this over a little bit. Where'd that go? There we go. I'll leave that a little bit bigger. Okay, so before we get on to eukaryotes, questions and clarifications on prokaryotes. This is your time to shine, Rose.
Oh, sorry, I thought my mic was on. I had a question on for the promoter and regulatory regions and the operator, where are both strands of DNA used, and depending on the direction of the gene, it uses the opposite strand? And
No. So, for example, when we're looking at, actually, let's bring up a better one than this. Here we go. So, for example, what I'm showing you here, let's move that over way a little bit. The direction of the gene in this case is from left to right. So what's called the coding strand, right? That's the bit that contains the information relevant to that gene, whether it's regulatory or coding, right, is the top one, right? Because it goes 5' to 3'. So that contains all the information for the gene. What you're probably thinking about is transcription, and transcription produces a copy of the coding strand, at least the bit that's transcribed. But it uses the other strand as the template, right? So because RNA can only be made in the 5' to 3' direction, and the gene reads in the 5' to 3' direction, you can't use the coding strand to make the RNA. You can only use the strand going in the other direction. So if you think about it, the only way you're going to get the same sequence, obviously with U's instead of T's, as the coding strand is if those bases are base paired during transcription with the opposite strand.
Oh.
Right. But only the coding strand has the information. So that's how you can have, say, it's not going to be the case in this gene, right? But actually, perhaps you have a shared regulatory region. I don't think this really happens in bacteria, but in eukaryotes it does. You can have a regulatory region here, right? And then you can have another gene going in the other direction using the opposite strand, right? So the regulatory region applies to both strands in that case. And so, whatever proteins are going to bind to that region to affect the gene going from left to right can also do the same for the gene going from right to left on the opposite strand. But the same principle applies. The coding strand is the strand which contains the information needed to make the protein and to control its production. So essentially, you'd have, if you got two strands like so, and you have a gene going this direction, gene going this direction. The top strand contains information for this gene. The bottom strand contains information for this gene. right? And typically they don't overlap much.
Oh, okay.
It's pretty rare that you'll find an actual gene overlapping with another one. Because if you think about it, for coding regions, whatever the amino acid sequence is for the gene on the top, right, the one on the bottom would have to have a amino acid sequence which is also contained in the information in that stretch of DNA, right? So you'd have very little ability to change that sequence,
Mm-hmm.
right? So it's kind of it's conceptually it's pretty unlikely to happen. Maybe it does, but it's not very common. But overlapping regulatory regions are fairly common. That's for sure. I actually found a few in some bioinformatics work I did for my own lab recently, which is pretty cool. Let's see who's joined. Hi, Alana.
Hello, how are you?
I'm doing well, and you?
I'm doing okay.
You feeling better?
Yeah, for the most part. My allergies are going crazy now, though.
It's the joys of San Antonio living, huh?
Yeah, so it's one after the other, and I'm allergic to oak, and I live around a whole bunch of oak trees, so it's not really helping me.
Oh, well, hopefully it will pass with time. But at least you have a snoring dog to comfort you, though. Actually, can you see? Let's see if you can see mine. There she is.
Aw.
Aw, Luna. Can you hear me? She's very suspicious of computers. For some reason, I don't know why. So Rose and I are just going through the prokaryote gene expression stuff, which we covered over the last two online sessions. Do you have any questions or things that you want to go over for that?
Not right now. I haven't been able to see the 17, the audio recording for that one yet, but I have looked through the slides, so I'm just kind of catching up right now.
Okay, well, if you do have questions, just bring them to, class next Tuesday. Or send me emails as well. That's fine too. It's just a little easier to answer questions like that in person rather than by email. But if you have stuff that you is kind of blocking you from getting onto the next kind of level of understanding that you want to get through kind of before then, just email me and I'll do my best to answer.
Alrighty, thank you.
Because it's pretty much, especially the triproperon is a little, I'd call it batshit crazy. Because it's complicated, right? So if you can watch the online session for Tuesday.
Okay.
I think it was Rose and Simon and I, but we had a pretty thorough go through of particularly this part, the attenuation.
Okay.
Because that's a little bit hard to get conceptually. There's a couple of things that you really need to understand before any of it makes any sense. So go through that pretty thoroughly, because that will trip you up otherwise.
Alrighty.
Cool. Okay, so what do we have today? Okay, so essentially what I want to do, this is the next two lectures pretty much this week, and Tuesday, next Thursday, we have, I'm not sure what one of the remaining PowerPoints will cover through that. I haven't picked which one yet, probably genomics, I would guess. And then the following week, so not next week, the week after that, Tuesday is either a catch-up or another topic, depending on what people want. And then the following Thursday is the exam. So essentially there's only 2 weeks of instruction left before your last genetics exam.
Are you talking about the final exam? Is that going to be through the whole year, or is it going to
No.
be just these last few chapters?
These last few chapters.
Okay.
So each exam is over the material preceding it. And that's the same for exam 4 too. There is no cumulative final as such.
Okay.
Yeah, because I don't know. I mean, if you don't know it from the first exam, you're not going to know any better by the time you get to the final, I don't think. The final does actually, hang on, let me just clarify that a little bit. Sorry, I'm just kind of remembering over again. Things fall out of my head fairly readily. It is semi-cumulative in a sense, right? It's made up of 2 sections. One section is for the last quarter of the course, right, which is what we're covering now since the exam 3. And then there's the other half, which is cumulative, but at a more superficial level. If you actually think about GenBio 1, when you had it with me, it's the same deal there. Do you remember that? Or was that too painful a memory?
Well, I didn't have class with you. I had lab.
Oh, that's right.
I had
I'm
class
sorry.
with Dr. Wise.
Okay, well, she would have done the same thing because
Yeah.
we'll do the same thing. So there's a half the exam. Essentially, you'll recognize it, right? Wink, wink. Which is over the topic as a whole, but it's not drilling down into like specific details. It's much more general concept and language, like vocabulary type stuff. And then
so it's just the basics that we should all know by now, and if we don't, we have no idea what's going on.
Pretty much, yeah.
Okay.
And then the other half is, you know, what you're used to by now, similar to exams 1, 2, 3, and so on. But I'll have to check. I can't remember from last time I did this, to be honest. I can't remember how many questions on each, but I'll check and get back to you to make sure. I'm just trying to think if it's longer or not, but I can't remember. I'll check.
Alrighty.
But yeah, it's, that cumulative part is in some senses more for our benefit than it is for yours. That should give you enough clues. Anyway, so okay, this is way more complicated than prokaryotes. So eukaryotic gene regulation, kind of in summary, is genes are off typically by default, and they're turned on when needed. Now, eukaryotes span in terms of complexity from protists, you know, think like yeasts and malaria. Yeast isn't a protist, it's fungus, but single cellular organisms, all the way up to, you know, blue whales, right? You know, large complex organisms that live in varying environments and things like that. So it's a bit tricky to talk about it because it's so much bigger range in terms of complexity than prokaryotes, for example, right? You know, bacteria and archaeas, either, well, you're an archaean or you're a bacterium. And if you're an archaean, you're an archaean. If you're a bacterium, you're a bacterium. And there are variations within bacteria, right? They do differ in terms of their habitats, right? But they don't differ hugely in terms of their gene regulation or their just general structure, right? And I'm sure microbiologists would be pitching a fit if they heard me say that. But they're relatively uniform, at least compared to eukaryotes. So most of the, in my opinion, and those of other scientists, but not everybody, most of the variation or the cause for that complexity is in control of gene regulation, right? Because you can, and this goes back to that C paradox or C value enigma. We have the same number of genes, give or take, as a soil nematode I work on in a lab, right? But we're obviously very, very different in terms of our complexity. Now, they're actually more complex than we are in many ways. They have more, as a ratio of their body, they have a far greater proportion of neurons, sensory neurons, than we do, right? They also have massive amounts of what are called nuclear hormone receptors, right, which basically detect signals, like, from externally and internally in different places. So they're far more complex than us in some ways, but they are far simpler than we are in other ways. And a huge chunk of that difference is really based on control of gene regulation, right? So that gives you a sense of how important and how varied that is. And there are multiple levels on which this occurs, and that kind of adds to the complexity. So every single level adds a different control, like a different dial or a different switch, to be able to vary the amount of active protein. Because really, in majority of cases, right, not all of them, what we're talking about is how much functional protein do you have present to do the job that that protein is supposed to do, right? If you think about it, we're going from DNA, and we've got a lot to do on that as well, to protein, and all the steps in between are subject to regulation, right? Subject to control. And that's kind of neatly summarized here in these two slides, right? Slides 2 and 3, which gives you an overview of those different levels of control, right? So at the beginning, you've got DNA. At the end, you've got function, essentially, right? And that, there's a lot to that, as you can see. So essentially, at the beginning, you've got your gene here, right? You're familiar with this concept already. But before you can even get to that point, you have to change the chromatin so that that gene is available, right? So the proteins can bind to it and transcribe it or activate transcription, right? So the very first level, you've got chromatin structure. And although this is a bit of a simplification, it helps, and it helps me as well, to think about this as a broad level control, right? So if you want to control of individual gene expression, typically, and this is again a bit of a simplification, as you'll see, is controlled by what proteins bind to that DNA, right? But you can control the access of those proteins to a wide range of genes by varying the chromatin structure, that immediate chromatin environment, right? And so although it's not always the case, it can be a very good way of turning off large regions of the genome, or turning it on as well, right? So it's kind of this idea of having transcriptional programs, right, or expression programs. Like, we want to express this, and this gene because they're important in this process, right? So we need to make sure that all of those genes are now available to transcriptional apparatus, right? That's kind of the idea. Similar in a way, if you think about it, to the coordinated regulation of prokaryotes, right? They just happen to lump all their genes into one single operon. Eukaryotes have a much kind of more nebulous way of doing that, but they do the same thing, essentially. Then the obvious one, which everybody thinks about, is control of transcription. All right, when you talk about gene expression, that's almost certainly, stop that, almost certainly what people think of is controlling whether or not that gene is transcribed, right? So that's the kind of classic sense. However, there's a lot more else going on, right? We've got RNA processing for a start. So each pre-mRNA is going to have multiple introns, right, which need splicing out. You can control how that's done, right? So that's one level of control. We'll touch on that. We're not going to spend a huge amount of time on it because it's really poorly understood, actually, to be honest. Then obviously that RNA is matured and then gets exported to the cytoplasm. Now we have a couple of other things going on. First, we have control of translation, right, which sounds a bit weird and wacky, right? But we can actually control via the 5 and the 3' UTRs, particularly the 3' UTRs, how efficiently translation begins. And we can actually block translation, right? There are small non-coding RNAs which will target the 3' UTR and actually prevent translation from occurring. We can also control how long that mRNA hangs around for, right? And this is actually a very important part of control of gene expression, because an mRNA, in the absence of anything stopping it, as long as it lasts, it will be translated, you know, over and over and over again, right? So if you want a, oh, you're a funny dog. If you want only a, let's say you want a lot of protein being produced, but you only want it for a short period for whatever reason, and then you want to turn it off. So you're going to turn on that gene and express and transcribe it at a very high rate. But as soon as you get enough protein, you want to shut things down. But now you have all this mRNA floating around being translated, right? You don't need any more protein. So the only way to stop that, you can't stop the transcription because that's already happened. The only way to really stop that is to get rid of the mRNA, right? And so that's the next step, right? So that's a very important part of shutting down gene expression, is getting rid of the mRNA, as well as obviously stopping transcription, right? You don't want to make any more of it because you don't need it. But you also want to get rid of the transcript that's already present. Then obviously you get translation, and that's where you get this active protein. And we don't stop here because there are two other things that can control its activity too. One is obviously if we don't need it anymore, then we degrade it, right? And so these proteins can be tagged. It's a process called ubiquitination. And they can be tagged to be destroyed by cellular machinery, right? Because these proteins contain resources in the form of amino acids, which are valuable and energetically costly. So we don't want that stuff hanging around when it's not needed. And you might actually not want that protein doing its job anymore, right? Because a classic example would be cell signaling, right? So quite often, if you want to reduce signaling between cells, you will take the receptor, the thing that detects a signal, out of the cell membrane and destroy it. So you reduce your ability to detect that signal. So you essentially turn down signaling, right? So that's one example. Or if proteins are damaged or defective or something. And then the final aspect is that protein has to be where it needs to be to do its job. And so if that's in the cytoplasm, then that's fairly straightforward, right? Proteins get made in the cytoplasm and then they're in the cytoplasm. But there are a lot of proteins do things in lots of other places, right? So these might be proteins that are involved in, oops, went too far, transcription, right? So they're going to have to be exported back into the nucleus. They may be hormones or something like that, which need to be exported out of the cell. And there might be hormones that need to be exported out of the cell only when they're needed. And so insulin is a classic example of that, right? So insulin is stored in small vesicles, export vesicles, in your beta cells in your pancreas, right? And so the insulin is not active because it is not flowing around in the bloodstream and it's not telling cells to take up glucose. And So the activity of insulin is controlled by whether or not it's actually exported out of the cell into the bloodstream. And until it gets there, it's not active. It doesn't do its job. Pardon me. But that also allows you to essentially do this whole process, right, of making protein, which takes time. And then you have a stockpile that you're able to release very quickly in response to, you know, a particular event, in this case, rise in blood sugar levels, right? So that's also another really important part of this whole process. Right? Anybody have any questions so far? Kind of a lot of information to take in. Nope. Cool. Well, I'm assuming you're still there. Oh, I can just keep talking to the screen. Okay, so what I'm going to do is jump around a little bit because through lackadaisicalness, I guess, I haven't sorted out the order of the slides in this PowerPoint. It doesn't completely follow that plan. So essentially, let's see what we got here. Yeah, there we go. Essentially, I'm going to start with chromatin. We'll bounce back to the other stuff. We'll probably get to cis-regulatory elements and stuff like that on Tuesday, because there's a lot of stuff going on. Give you a little, say hello to my dog again. Isn't she beautiful?
Aw.
Look at this squish. Yeah, see, your dog approves. She's a cutie pie. She's a complete wuss as well. Yeah, she's scared of all kinds of things. Anyway, so okay, so essentially this is again going back to chromatin structure, proteins, histones bound to DNA. And there's a lot of stuff that can go on here. We touched on a little bit of it earlier in the class when we talked about how tightly bound DNA is to the histones, right? What we're going to do now is expand on that, right? Because there are other ways in which you can control how accessible DNA is to proteins that will turn on gene expression via chromatin modifications. There are various different ways you can do that. So essentially the idea is that if DNA is tightly associated with histones, it's not able to be expressed, right? So compact chromatin, which is essentially what that means, is associated with transcriptional repression, right? And the reason for that is simply a physical one, right? The proteins needed to turn on gene expression, which we'll get to later, such as transcription factors, RNA polymerase itself, things like that, they occupy physical space, right? So there's a physical occlusion of DNA, right? It kind of just physically is not accessible. And the other part is that these proteins that turn on gene expression are DNA binding proteins, right? They have specific motifs and parts of their structure which recognize specific DNA sequences, right? And those DNA sequences have to be in a particular shape to be accessed or read. And so if you look at just this diagram here, for example, right, that transcriptional activator protein binding site, or this TADA box protein binding site, these are all DNA regions which are recognized by proteins that recruit RNA polymerase to the promoter, right? And so those have to be accessible for those proteins to be able to bind them. And so being wrapped tightly around a histone complex means that they are not available for binding. So that whatever gene is, downstream of these, right, coding region that way, let's say, cannot be turned on because those proteins cannot bind, RNA polymerase cannot bind, and so that gene is off, right? Hang on, this isn't. So that's just a general general concept, which I'll let you kind of read through that. So essentially, there's different ways in which you can control how accessible DNA is to gene expression, right? And these are called epigenetic modifications, right? They're called epigenetic because they're changes to the ability to for DNA to be expressed, right? Or essentially for a particular function to occur, from a piece of DNA, but they are not changes to the DNA sequence, right? So you can change how much, whether or not even a gene works, right? Through these modifications, but you don't change the sequence of that gene. And also those changes, those essentially a kind of non-genetic, it's not really the right word, non-DNA sequence changes are heritable. So part of what makes you who you are as an individual, obviously you inherited that from both your mother and your father, but it's not entirely based on the DNA sequences that you inherited from your mother and your father. It's also based on what epigenetic marks did you inherit, in that first cell fusion between two gametes. And so essentially there's two different things that you can do. You can change, well, that's a big thumb. Let's move it back here a little bit. You can change the histones, not the histones themselves. You can modify the histones. There you go. That's a better word. You can modify the histones, and there are two different modifications you can do. There's methylation, which is typically repressive, right? So that's adding a methyl group to the histones, and there's also acetylation, which is typically a positive effect, right? Promotes gene expression. You can also have methylation of DNA, right? Which, as with histones, is generally repressive, and this is called cystone methylation. So those are two general things that you can do, but there are also there's another kind of category which is really kind of a more generic, essentially, in a way, and they're called chromatin remodeling complexes. So essentially, this is whenever I talk about this, I always think about an abacus. So if you think about the rod and an abacus, you both know what an abacus is, right? Hope I'm not. They're probably like now childhood toys. They're really useful for like counting things. No? Anyway, basically wooden frames with metal poles, and they have beads on. They're used for counting.
Yes, they used to be in the doctor's offices, right?
Yes, that's right. And kids' toys as well, which is kind of crazy. There are actually fairly advanced ways of doing, like, calculating stuff. I've just never figured out how. But anyway, essentially, chromatin remodeling complexes, their job is to kind of shuffle those beads around, right? And sometimes just take them out completely to be able to change the accessibility of that DNA to various proteins. Right, and those obviously take up, they consume energy. And so there are lots of different levels you can work at here, right? Chromatin remodeling complexes are, can affect large regions, potentially. Histone modifications, likewise, or they can affect just very specific small regions, like for a particular gene, just one gene. And DNA methylation is typically gene specific, right? So this is, again, more specific further. And DNA methylation tends to occur in promoters, right, of genes, much less so in coding regions. So it's definitely a specific regulatory process. Where are we at? Okay, so essentially there's lots of different things that can be done, right? So what we're seeing here is essentially you can, because if you remember, there are these histone complexes. You have a nucleosome, which is the 8 histone proteins, 2 and a bit wraps of DNA, and then you have a bit of space, right? This linker DNA, like 50 bases, give or take, about a third of the DNA that's wrapped up in the histone. And so what chromatin remodeling complexes can do, or CRCs, right, just take less time to say it, is they can rotate those histones, right? So essentially, if you have a histone complex or nucleosome here, right, the DNA stays still, and then the histone will rotate along, so the bit of DNA that was once wrapped around it here is now free, right? And so if you imagine there's all these histones along the DNA strand, each with gaps between them, if you have a particular region which is important wrapped around one of those histones, you can potentially just rotate that histone out of the way, and then that region will now be exposed and it'll be accessible to whatever protein needs to bind. In this case, it's the Taddabox binding protein and transcriptional activator protein. So you can actually see, let me just double check, there you are. So for both of these, and this kind of goes back to, let's do this carefully, this one here, you know, both of those binding sites are obscured by histones, right? They're bound. Oh, the cat's coming down there. Oh, got another cat. Let's see if you can see. I don't know if you could see her. Oh, there's a cat on the stairs. He's probably complaining about something. They're very wary of the dog because our dog's a bit of a jerk to the cats. Oh. Anyway, so if you can see here, those binding sites are obscured. Now with the use of those CRCs, those histones can be rotated down the strand a little bit, and now those sites are exposed and accessible to those proteins. And so what quite often happens when that occurs is that these 2 proteins, the TAP and the TBP, they recruit RNA polymerase, and what RNA polymerase can do is it can actually kind of plow off the histones ahead of it, right? So it can actually force that that DNA to unwind off the histones, and it can force the histones off ahead of it, a little bit like a like an icebreaker, right, going through the Arctic, you know, forcing the ice apart ahead of it. So as long as those sites are available, RNA polymerase can actually get in and you know, further clean that DNA strand off. And obviously, once RNA polymerase has gone through and transcribed the gene, depending on how active that gene is, then those histones can just reassemble afterwards. Okay. What can also happen, what's down here? There we go. Is those CRCs can simply remove histones from the DNA, right? So if you think about this is, let's say, tightly wound chromatin at the top, euchromatin. If you want to get access to that DNA, it would just help if there are no histones present. So those CRCs can not just rotate or slide histones up and down the DNA strand, they can just kick them out. And that is obviously a much more effective way of promoting gene expression, because now that whole bit of DNA is accessible. So that's CRCs. Anybody, either of you, got any questions about that?
I don't think so right now.
Okay, cool. I mean, it's fairly, straightforward in a sense, right? How they work. It's pretty easy to visualize. Okay, so what's next? Sorry, I got kind of allergies, I think.
So I guess I did have one question. You says that they can unwind and remove the histones when they unwind, or I'm sorry. Let me think about my question for a second. Do they don't always remove the histones, do they? Sometimes they
No.
just move it to a region where they can be accessed.
Correct. So you can just slide them along, like on an abacus, essentially, right? So if there's a particular bit of DNA which is obscured by it or wrapped around by a histone octamer, then if you want to access that bit of DNA, you can either remove the histones completely, right, which is an obvious way of doing it, or because there's a gap between each nucleosome, you can slide those histones along a little bit. It's like if you yeah, so essentially like a bead on a necklace, essentially. You can just kind of push them along a little bit. But because the DNA is wrapped around them, they're not slid so much like beads are, they're rotated.
Okay.
So essentially a different bit of DNA is now wrapped around the histone than there was before.
Yeah.
All right. Okay, so that's kind of broad brush stuff. You can also get into specific changes to histones themselves, and this really comes down to how histones interact with DNA. So, and there's actually a histone code, right? And if you ever get to read any papers about stuff like this, and I've read a few, it's like reading another language. They will talk about which histone is being modified and which amino acid residue on which histone is getting changed. So there are 4 pairs of histones, 2A, 2Bs, 3s, and 4s. And each of those has tails. And so the tails are really the big deal, right? That's where all these changes occur, right? The actual bodies of the histones that are inside the nucleosome are not modified at all. It's always the tails. The tails are modified because of their interaction with the negatively charged backbone of DNA, right? And so these tails on histones have a whole bunch of typically lysine amino acids, and lysines are they're basic amino acids. Get off. So they have a positive charge typically, right? And so if that those lysines are just present as is, right, that histone tail will bind tightly to the DNA through just an ionic interaction, right, forming an ionic bond. So essentially what you can get now is 2 modifications to those tails. You can methylate them. I'm not actually 100% sure what methylation does, to be honest. I don't know very much about histone methylation in general. But if those tails are methylated, DNA is bound much more tightly, right? And there's also an interplay between methylation and acetylation. Now, if you want to kind of open up that DNA and unwind it, right, make it more accessible and not so tightly bound to those histones, pardon me, the best way to do that is, if you can imagine, there's all those lysine residues, right, on those tails, positively charged amino acid residues. What you can now do is you can add an acetyl group to those lysines. And what that does is it basically removes that positive charge, right? So the outcome of acetylation, right, not methylation, which again, I don't really fully understand, but the outcome of acetylation is to make these tails neutral. And if they're neutral, they have, or more neutral, right, less positive, they have a much weaker attraction to DNA, and that DNA then, it doesn't get removed completely, right? It's more that it kind of, instead of being tightly bound, it then just kind of relaxes out. And so again, that DNA is then more accessible, more available to other proteins that want to bind to it, right? So when we talk about histone modifications, we're typically talking about methylation, which is associated with transcriptional repression, and acetylation, which is associated with transcriptional activation, right? And there are enzymes which will switch back and forth, right? There's what they called HATs and HDACs, histone acetyl transferases and histone deacetylases, right? So the various activity of those two proteins in a particular region will dictate how acetylated those histones are, which then dictates how transcriptionally active that DNA is, right? Because one thing to remember is that all of your genome is associated with histones, right? Every single bit of DNA that you have, other than mitochondrial DNA, in your cells is in this form, right? One of these two forms, let's say, right? And so there's no just open stretches of DNA. So whether or not it's transcriptionally active or repressed, it's always going to be associated with histones. And so how those histones interact with that DNA dictates how, whether or not that bit of the genome is going to be expressed or not. Any questions? It's kind of straightforward, but it's massively complex. Kind of at a superficial level, it's straightforward at least.
I had a question on slide 16. Like, it shows the structure of the DNA strand. What is it necessarily trying to show? I understand that when you add or for histone acetylation, the positive charge becomes neutral, and therefore it lessly tightly bound around the DNA. But what is it trying to show in the picture of the DNA?
Right. So if you look ever so closely, you see that O just above my cursor.
Mm-hmm.
What charge does it have?
Negative.
That's what it's trying to show you.
Okay.
So, and this gets back to the whole idea of you can't change the charge of DNA.
Mm-hmm.
So if you want histones to bind less tightly, the only thing you can change is the charge of the histone, right? Because the charge of the DNA is intrinsic to its structure,
Mm-hmm.
right? So if you want this to be bound less tightly, you can only reduce the charge of the histones to reduce that attraction between the two. And that's what acetylation is all about.
Oh, okay.
And there are multiple, obviously multiple there are 8 tails per octamer.
Mm-hmm.
And each one of those has multiple lysine residues. I think they're typically lysines from memory. And so depending on which one of those is acetylated, how many of them are acetylated, right? It's not an on or off deal. It's, again, a dial. And those different residues get acetylated in response to different cues. For example, maybe smoking, if people still smoke crazily enough, will result in acetylation of particular lysine residues, but not others. You know, elevated blood sugar levels for long periods might result in acetylation of a different group of lysine residues, right? So there is very much a histone code of which residues get acetylated in response to which, you know, signals, essentially. Like I said, it's astonishingly complex. It really is. It's just mind-boggling. Like the level that we're teaching at is like the most complex level that I've ever wished to go into personally. But that's again, it's one thing that you'll learn as you go through your degree, and you know, you learn more about biology, is that we know actually very little about a lot of biology. And this is a fabulous example of that. You know, we know some of this, but it's something that's almost beyond our ability to understand, in a sense. It's so astonishingly complex. So anyway, there you go. So yeah, that's what that particular slide is all about. And again, I'll look into what histone methylation does, because I don't fully understand it myself. So I'll dig into that, see if I can figure it out a bit better for next class. So yeah, there you go. A good example that I don't know everything. There's an awful lot that I don't know. I'm not sure if that's supposed to be comforting or scary or not, but anyway. Any other questions on histone modifications? No, cool. Okay, so next one is, kind of covered that already, DNA methylation. So this is actually really interesting because I actually had a thesis student once, my previous job, who, her project was correlating DNA methylation patterns with cancer. She did like a summer research project or something like that. And DNA methylation is typically repressive, right? It will shut down gene expression, and it almost always occurs in the promoter region of a gene, right? And what you'll tend to find is, at least in cancer, there's a bunch of essentially anti-cancer genes, right, which are involved in controlling cell cycle progression or promoting apoptosis, or you know, a bunch of different processes, but particularly involved in cell cycle checkpoints. And what they've found, and the student was part of that, is that a lot of the genes that are involved in suppressing cell division and cell cycle checkpoints are heavily methylated in people with cancer. And so it appears that one of the consequences of cancer is that it actually inhibits or shuts down, and it's kind of hard to know cause and effect here, right? The genes which would be responsible for controlling it. So it's almost like, you know, it's gone around the back and like shut down all the security cameras and stuff like that. So, you know, the security guards can't see it. And so DNA methylation is a really interesting thing. But again, now we're more gene specific, right? So this isn't necessarily a promoter specific, like for a particular gene, but it tends to occur at the gene level rather than at the, you know, the large scale level that chromatin remodeling and histone modifications tend to act at. And so what DNA methylation is about is that these cytosines, right, and you have what are called CpG islands, there we go, which are CG dinucleotides, right? So wherever you have a CG in a stretch of DNA, and you tend to find a lot more of those in promoters than you do in coding regions for whatever reason. Wherever you have a CG, the C or the cytosine can be methylated, right? And so if that C is methylated, and again, it's not an on-off, it depends on how many of those CGs or those CPG islands are methylated, right? So it's again as a dial, it's a variation. Depending on how many of those Cs are methylated or how heavily methylated the promoter is, can affect how readily that gene is expressed, right? And if it's heavily methylated, it will typically be transcriptionally silenced. It'll be shut off. And even if proteins want to bind to that regulatory region or that promoter, they cannot, right? So even if there's a signal saying, hey, we need this gene product, come on, we need this protein. If that promoter is heavily methylated, it won't happen, right? Even if there are things, even if there's no chromatin present, even if there's plenty of proteins that want to bind to it present, it won't happen. And that's partly because those methyl groups, they kind of change the shape of the DNA. And so a lot of those transcriptional activator proteins or transcription factors won't physically be able to bind to that DNA. Also, that change in shape is recognized by repressor proteins, which will kind of similar to if you think about LAC-I, the lactose repressor. They'll just physically bind to that promoter and it will stop other proteins like transcription factors from binding. So it's a pretty effective way of shutting down the expression of a gene. And again, these are inherited both from cell to cell and from, parent to child, because these marks are copied every round of DNA replication, right? So and this is actually, do you remember when we talked about mismatch base repair right after DNA replication?
Yes, I remember.
Yeah. So this DNA methylation is how the mismatch repair mechanism recognizes which is the original strand and which is the strand with the mistake in, right? Because here we go. Essentially, during DNA replication, only the template strand will be methylated. The newly synthesized strand will not, right? Because it hasn't had a chance to be done so yet. So that fresh, squeaky clean DNA strand, that new strand, that's what's kind of known is new, right, for repair of any mismatches. However, after time, after DNA replication has finished, or at least has moved on from that point, then these methyl transferases will come in and they'll copy the marks onto the other strand that were originally there in the original strand. If you look up here, they will then come in and they'll restore those marks. And so when you inherited your parents' chromosomes, right, and their selection of alleles, you also inherited their methylation marks, right? So even though that change isn't necessarily permanent, right, doesn't necessarily mean that your children will inherit the same marks. Most likely they won't. They'll inherit some marks that are the same and some that are different. But they are heritable. It is heritable information, which is kind of crazy when you think about it, right? The whole concept of inheritance is a much more complex one than we've been telling you. So hopefully you don't feel like we've been lying to you all this time or something. Because we haven't. We just don't want to blow your brains out too quickly with too much information. It's funny, actually, Just a quick joke. Let's see if I can open up something. Where are we at? I had a laugh with my astrobiology class yesterday. Here is it. Do you recognize this species of frog? Get it?
That's good.
I actually had one of my students, he Google searched that. He's like, I've never heard of a genus of frogs called Kermiticus. It's because it looks like Kermit the Frog, but frozen. Anyway, so there is no frog called Kermiticus chileus. I just made it up. So I told them they can't trust everything their professor tells them. Which I thought was pretty funny. So again, right, we haven't done any of this stuff on cis-regulatory elements because it just doesn't really fit in the sequence. Again, I need to go in and sort these PowerPoints out a little bit. But we'll get to that next class. But just to leave you with, actually, we'll probably go through that next class too. But just a little kind of like a whisper in your ear, essentially. These two, DNA methylation and histone methylation, interact. So there are positive feedback loops that occur. And so if you find quite often, not always, but quite often, you can have the case where methylation groups on DNA recruit, let's say, histone deacetylases, which then remove acetyl groups from the histones and further shut down gene expression. So if you think about it, if you want to be like absolute, this gene shall not be expressed, right? Say your X chromosomes, right, in the two of you, right? Because I only have one X chromosome in each of my cells. But in each of your cells, you have two X chromosomes. And one of those X chromosomes is shut off completely. It's completely inactivated, partly because of issues of gene dosage, right? But essentially, if you want to really definitively shut down gene expression from a particular region or even a whole chromosome, these feedback loops kind of start playing an important role. And so you can get both directions, both between histone methylations then lead into more DNA methylation, and DNA methylation leads into more histone deacetylation or histone methylations too, right? So there's feedback between those two. It's like, and it's a positive feedback too, until things are completely shut down. But anyway, we'll go through that in a little bit more detail next class, because I think you've probably had enough for now. So yeah, if you got any questions, write them down, bring them to next class. We'll have a little bit of time before we get on to the rest of this and cis-regulatory elements and stuff. Yeah, for the rest of the class. Okay, so yeah, stay safe. I hope your allergies get better, Alana. It can't be a lot of fun.
Thank you. I actually did have a question about lab because I missed yesterday.
Okay, that's fine. There wasn't an awful lot that happened yesterday. What was your question?
Well, it seems pretty straightforward. I looked through all the videos and everything. I downloaded what I needed to. Just in the folder in Blackboard for the lab, there's the zipped file, which obviously we need that. But there's a couple other things that I wasn't sure why they were.
Some of them are just relics from previous labs or from previous versions of this lab that we would have used if we were doing this in person. So let's just go have a look at that fucking yank it off of there. Thing that annoys me about, there we go. That's the one we want. Let's see if this will let me in. Okay. What'd you do that for? Okay. Bum, bum, bum. It's thinking about it. Okay, so this one at the top, that's your worksheet that you need to work through. This is your written descriptions of what you need to do, and that dovetails really nicely with all those videos that Dr. Page produced. And this is the zip file of your DNA sequences. You don't need to do all of those, just pick a handful of them.
So
right.
About how many would you
5 or 6 should give you a good readout. You don't need to do more than that. I think there are 30 in there.
Yeah.
Yeah, so just randomly pick whichever ones you want to do. It's not, you won't be, the questions in here don't rely on you showing like a picture of, oh, I've done like all 30 or something. They're about answering what you find from doing that. And so I think you only need 5 or 6 to be able to answer that question more than adequately. I'm not going to tell you what that is because you have to go download it yourself. You can kind of figure that out. These are old sequences, and this is the wild type sequence. Actually, this is a readable file. I'm just curious what this. I should just open this up from my folder on my own computer, shouldn't I? So this should be the, because you're instructed to download the mutant sequence, right? The allele. Yeah, so this is the wild type sequence for the yellow gene. And so when you do your alignments, try, if you're looking for a specific difference between, and you should be, between your yellow sequences, the sequence files that you've been given to analyze, and the reference sequence, the best reference sequence is actually the wild type sequence, because that should show you there should be one base difference, and it will show you exactly where it is.
Okay.
If you use the yellow reference sequence from Flybase or NCBI, that will have that change in already, so it will match perfectly with all of your other yellow sequences which come from yellow fires. Most likely, I'm not actually I don't know. Isn't that weird? I'll have to double check with Dr. Page, because I can't remember if he said that those yellow sequences came from yellow flies or not. I think they did. I'll double check, and I'll send that announcement once I've got confirmation from Dr. Page. Yeah, so that's what those other files are for in here. And there's a couple of papers in here as well. These papers
I
which are
saw
about
them.
that. You don't need to worry too much about that. That's more for if you're doing the actual full fly report, which we're not doing anymore.
So all we're doing is are we doing a results section? Is that what it is?
Correct.
And that's it? We're not having any lit review at the beginning, nothing?
No, if you want to add just a few words as to purpose, and then in your results section discuss, make a statement about what you found or not, that may help. But the actual rubric isn't going to grade you on that.
Alrighty, sounds good.
Yeah, so there was supposed to be the methods and protocol section as well, but because of the disruption from the switch to online teaching, that was dropped.
Okay.
Any other questions?
That was about it.
Cool. I will double check with Dr. Page, and I'll get back to you about the source of those yellow sequences. He may have put it in his email, but I can't remember it off the top of my head.
Alrighty.
Cool. Rose, you good?
Yeah, I'm good.
Cool. All right. So take care. See you next Tuesday. Have a good weekend.
Alrighty, you too.